This is Biology 1, Chapter 10, Part 5 of Sexual Reproduction and Genetics. In this portion of the video lecture, we will be discussing gene linkage and a condition called polyploidy. We've talked about why meiosis is so important for genetic variation. Remember, during meiosis, we have crossing over, which happens in prophase 1. And we also have the principle of independent assortment that was developed by Gregor Mendel. Both of those things together give us what's called genetic recombination, which is genes produced by crossing over and independent assortment. So basically, you can think of genetic recombination as every possible allele that exists between two sexually reproducing organisms. So every possible combination that there could be in their genome that would be represented in their offspring. We can calculate that by taking a look at a very simple equation of 2 to the nth power. Remember, we are diploid organisms. We have full sets of chromosomes. So we have the number of chromosome pairs that is represented by that nth power. So for example, in people, since you need a male and female, it takes 2 to tango, you have 2 to the 23rd power, and you multiply that by 2 to the 23rd power, and that will give you an awfully big number. Approximately 70 trillion different combinations of genes can exist between two different people. Basically, if two people could have 70 trillion children in their lifetime, the 70 trillionth and 1th kid would be an exact duplicate, an exact clone of kid number 1. Now, that's why we have at least 7 billion people on the planet right now, and we are all genetically different. Gene linkage is a phenomenon that was more recently discovered in the history of genetics through taking a look at how genes are expressed. It was realized that some genes are so close in the chromosome that they end up being inherited together. And we can actually now, with the technology that is available, use what's called a chromosome map. And you can actually sequence the position of genes on the chromosome. So that uses crossover data. So the more times genes cross over, that means you're going to have a lot of sequences on your gene. So the medical community uses that to figure out where genes are on chromosomes. Polyploidy is a condition in both plants and animals where you get an extra set of chromosomes. Remember, poly means many, and ploid is referring to the chromosome. So you get extra whole sets of chromosomes. This is very common in plants, especially plants that we use for crops, things like corn and soybeans, strawberries. Those can be polyploidy, and it does not make a difference to that plant. In some cases, polyploidy can actually be lethal in humans. We will talk about some non-lethal forms of polyploidy in our next unit, but know that polyploidy is extra sets, and it's very common in plants to have extra chromosomes. This has been part 5 for chapter 10 on sexual reproduction and genetics.